Hello, good morning, Miss Paila. My name is Jamal, surname Dakwit, and this is the oral presentation part of my major project about the food microbiology research portfolio. And the section that I chose to present as my oral presentation part is the section D. So, in my general introduction about food microbiology, I just want to elaborate a little bit about food microbiology. So, food microbiology is a vital discipline within applied science that examines the interactions between microorganisms and food systems. Microorganisms, including bacteria, fungi, viruses, and parasites, play both harmful and beneficial roles in the food chain. On one hand, they contribute to food spoilage and cause foodborne diseases that threaten public health, leading to significant social and economic impacts. On the other hand, selected microbes are harnessed in fermentation to produce valuable food products such as yogurt, cheese, soy sauce, and traditional fermented foods. This dual nature of microorganisms makes food microbiology is a field of immense importance for ensuring both food security and food safety. So, on my oral presentation, the section D, which is about the microbiology in food fermentation, and my focus part is about the yogurt production. So on the yogurt production, I give a title of my own, which is the role of microorganisms in yogurt production or yogurt fermentation. So today I'm presenting on the microbial processes behind yogurt fermentation. So one of the world's most widely consumed fermented dairy products. So fermentation not only improves food safety and preservation, but also delivers health benefits such as improved digestion and probiotics. Next, the microbes involved in yogurt fermentation are two types. Two main lactic acid bacteria are used, and they are Streptococcus thermophilus and Lactobacillus delbrueckii sub sp. So Streptococcus thermophilus begins fermentation and Lactobacillus delbrueckii sub sp continues acidification. So these microbes convert lactose, which is the milk sugar, into lactic acid, lowering the pH and forming the yogurt texture. In the fermentation process, the milk is pasteurized at 85 to 90 degrees Celsius to kill the pathogens and denature proteins. It cools to 42 degrees Celsius, and then the lab starter cultures are added. The fermentation occurs for 4 to 8 hours until pH drops to 4.5 and let cooled and stored under refrigeration. And also in the yogurt fermentation, it has the health benefits. And the health benefits of yogurt contains live probiotics that improve gut microbiota, aid lactose digestion for intolerant individuals, and it may also reduce cholesterol and improve immunity, and it is easier to digest than milk due to lactic acid fermentation. In yogurt, the food safety considerations are also vital. So, in food safety considerations, we must use only pasteurized milk, and we must stick to the temperature control, which it is necessary to avoid pathogen growth, and also the hygienic handling prevents post-processing contamination. And we must also at lab themselves inhibit sporage microbes via acid and bacteriocin production. So the infographic poster of how yogurt is made is already printed as my hard copy and submitted, so you can check it there for your verification. So to conclude, yogurt is an excellent example of how beneficial microorganisms can transform raw food into a safe, nutritious, and valuable product. Finally, as a future food scientist, it is vital that we understand the microbial processes involved in both food safety and functional health. That's the end of my presentation, and you have a wonderful day. Hi, thank you.